Good evening and welcome to the live practice session for the NPTEL course on Applied Environmental Microbiology. So this will be the week 6 course, and in this session we'll be talking about symbiosis, about the virus, and some amount of applied environmental microbiology. So this was the course content that was discussed in the previous lectures as well. So in previous live sessions we have seen some of the basics, and in this week we'll be seeing about the relationship between microorganisms, the virus part, and some amount of application oriented stuff of the course. So the first question is, in a microbial consortia, so as always we'll solve some questions only. So in microbial consortia involving so welcome to the session again. So we are just starting now. So the first question for today is, in microbial consortia involving green sulfur bacteria and motile non-phototrophs, the primary ecological advantage of the motile partner is which of the following? Uh, so that is the first question for today. As stated, we will talk about the different kinds of relationship among microorganisms in the environment. So the set of questions that follows will be based on that. And apart from that, if you have any doubts from any of the lectures or from previous live sessions, you can also post that in the chat box. In the meantime, you can also try answering this question. I hope I'm audible and the screen is visible. If the audio and the presentation is visible, please let me know in the chat box that it is fine. Please let me know if I was audible and the screen is visible. physiological advantage of the motile partner is? Uh, so there is a consortia which has a green sulfur bacteria and motile non-phototrophs. What is the benefit from this association for the motile non-phototrophs? That is the question. Okay, so the answer for this question is that position optimization along with physicochemical gradients. Okay. So in a phototrophic consortia, like what happens is usually a green sulfur bacteria and motile non-phototrophs. Uh, like typically some proteobacteria like microorganisms are in consortium. So here the green sulfur bacteria is immotile, and again it is a anaerobic photolithotroph. Like we have seen about green sulfur and trophic sulfur bacteria in the previous session. So a green sulfur bacteria is a anaerobic and it is a photolithotroph. So it requires both light and sulfide for its growth. Okay, but it cannot move. So what happens is this motile partner helps the uh, green sulfur bacteria to move. So the advantage for the motile bacteria in this case is uh, it is a heterotroph and it cannot fix its own carbon. So what happens is it relies on the autotrophic green sulfur bacteria for essential organic compounds. So this way both of them are benefiting each other. So the next question is green sulfur bacteria in stratified lakes are typically restricted to anaerobic zones because of what? Just now we saw that green sulfur bacteria is anaerobic. So what is the primary reason for which it is restricted to anaerobic zones, or why is it anaerobic? Audio is clear, right? Can someone just post in the chat box if the audio is clear? And also the screen is visible, the question is visible. Just give a confirmation in the chat box and then try answering this question. And also if you have any doubts, please let me know. Thank you, Jeetu. are usually restricted to anaerobic zones because they possess oxygen-sensitive photosystems. So we all know that green-sensitive bacteria are obligate anaerobes, which means they have to compulsorily be in anaerobic condition. If they are even exposed to a little amount of oxygen, they cannot survive in that environment. And also, their photosystems are very much oxygen sensitive. Their nitrogen fixing enzymes are also very sensitive to oxygen, which means in the presence of oxygen these enzymes will not work, and if they are not working, then the bacteria cannot perform carbon fixation or nitrogen fixation, or energy generation. Any of it, all of it is going to be stranded, because all these key enzymes and photosystems are very much oxygen sensitive. So that is the whole reason why this green sulfur bacteria are restricted to anaerobic zones. And one more interesting fact is that this green sulfur bacteria also performs the anoxygenic photosynthesis. So oxygenic means it will generate oxygen. So in photosynthesis we know carbon dioxide will be taken up, while oxygen will be produced in the process. But the process that green sulfur bacteria does is an anoxygenic photosynthesis, where they do not produce oxygen as byproduct, because they are usually reduce sulfur compounds, like sulfides are required for green sulfur bacteria growth, right? So this sulfide, they utilize the sulfur and water as well. So next is the easy question: what is a lichen? So there are two organisms that are involved in this symbiotic association. You just have to name what they are. What are the two organisms? are more fungi. So this gives the uh, arena for nutrient uptake, and as well as this forms the thallus or the supporting structure. Along with this, there exists algae or cyanobacteria, which obviously performs the photosynthesis, and it produces carbon for the fungi to consume. So this is the symbiotic association. Symbionts in the sense both the organisms are benefited. So here the organisms that stay together are fungi and algae, and both these organisms are benefited from each other. Okay, so based on whatever I told in lichen symbiosis, the photobiont primarily contributes to what? Exactly. So the contribution of the photobiont. So in this case, the term photobiont refers to the photosynthetic organism, which is the algae or the cyanobacteria. Okay. So the role of the photobiont is to fix carbon via photosynthesis. So it is responsible for photosynthesis, thereby it generates some organic compounds which can typically be used by the fungal partners. Okay, and when we talk about the structural protection, the fungi is the major component that gives this strength, or which builds the physical framework of the lichen, and you call it thallus. Okay, so this is very essential because it protects from desiccation, like it keeps the surface damp, and it prevents dryness, and also it gives some structural stability for not being physically attacked, and also protection from extreme light, temperature kind of environmental factors as well. And when we talk about nitrogen mineralization is kind of a specialized um, phenomenon that may or may not happen, but usually in lichen symbiosis, uh, nitrogen mineralization is not a very uh, primary aspect that you look into. And when we talk about spore disposal, we know that the fungus or the microbiont part is going to produce spores for further propagation of this association. The answer here is in the photobiont component in lichen symbiosis contributes to carbon fixation via photosynthesis. So the next question is, in rumen fermentation, hydrogen removal primarily occurs through which of the process? Exactly. So the answer is methanogenesis. So when we talk about the rumen of animals, uh, there happens this breakdown of complex carbohydrates, and after this breakdown, which is the hydrolysis, there comes the next step called fermentation. And we all know that fermentation is the process that is going to yield us products like acids, alcohols, etc. Right. So when we talk about rumen fermentation, the primary product is going to be volatile fatty acids, like those that we call as acetate, butyrate, propionate, etc. All these are volatile fatty acids. They are short chain. So these are the primary products in the rumen fermentation. So along with this, as a byproduct, you also get hydrogen, and it is very very important that the hydrogen is being removed from the system. When you talk about the biochemical perspective of this process, a higher production of hydrogen is going to kind of feedback into it. So this process is not thermodynamically favorable. The fermentation is not thermodynamically favorable when the hydrogen concentration is very high. So initially there won't be much hydrogen concentration, but when the fermentation progresses, hydrogen production is there. So further fermentation will be inhibited by the hydrogen produced. So in order to overcome this thermodynamic limitation, the hydrogen must be consumed from the system, and the primary pathway by which the hydrogen is consumed is methanogenesis. So in the rumen environment, we also have methanogenic archaea. So we have seen about this prokaryota, archaeota in the first week session. Eukaryota, prokaryota, and archaea, right? So archaea can thrive in extreme environments. So in our case, they are going to be methanogenic archaea, which will generate methane. So how they generate methane is you have both carbon dioxide as well as hydrogen. So both of these are consumed by the archaea, which in turn converts them into methane, which is CH4. There can be sulfate reduction, nitrification, denitrification, and all, uh, but the role is very, very, very limited. The predominant or the primary pathway for hydrogen removal is methanogenesis, and the reason for hydrogen removal is a higher amount of partial pressure or the concentration of hydrogen in the system will further inhibit the fermentation process. match the following quiz. Okay, so the answer is A3, B2, C1, D4, and E5. So you're absolutely right. So when we talk about lichen, so we have just extensively seen that it is the association between um, fungi and algae. So here the fungi depends on algae, which produces organic compounds as a result of photosynthesis, and the algae depends on fungi for structural stability and nutrient uptake. Okay, so that is a kind of a photosynthetic mutualism. Photosynthesis happens there, and both organisms are mutually benefited. So, as the term can easily suggest, lichen stands for photosynthetic mutualism. When we talk about root nodules, so root nodules are generally present in leguminous plants where there are certain um, bacteria that are present in the roots. So they generally perform nitrogen fixation. So that is why root nodule matches with nitrogen fixation. When we talk about rumen, we just saw that there is going to be methanogenesis or methane production for the removal of hydrogen. And when we talk about hind gut fermentation, so hind gut fermentation is matched with fecal fermentation. So fecal fermentation is the phenomenon in which in some animals the entire digestion process does not take place at the starting or in the front act of the digestion process itself. Okay, so this cecum is located at the junction of the small and large intestine, and at this point there are some enzymes that can break down complex fiber and plant material which were not digested in the small intestine. And this process we call it as cecal fermentation, and the microbes involved here are hindgut fermenters. So that is the match. And when we talk about green sulfur bacteria, we saw why a green sulfur bacteria is anaerobic, because its photosystem is sensitive to oxygen. So it it can only exist in anaerobic condition, and that is why it is obligate anaerobe. And regarding phototrophy, yeah, it has photosystems that can harvest even dimmest of light, so it's that powerful. And it's a photoautotropic microorganism. So this is the right match. Another match the following question here about foregut and hindgut fermentation and other aspects. Foregut fermentation is pre-gastric fermentation, and hindgut fermentation is post-gastric fermentation. So as the name suggests, it's very easy to place both of this. So in foregut fermentation, the easily fermentable food generally gets fermented, and there is production of fermentation products, whereas the complex fibers and stuff that are present further goes to the hindgut fermentation, which we saw in the previous question as well. And when we talk about uh, coprophagy is ingestion behavior, where the animal tries to re-ingest or take up its own feces or any other species. This behavior is kind of common in some classes of animals, and this can sometimes increase the microbiota but can also act as medium for pathogen transfer as well. So coprophagy stands for re-ingestion behavior, and stratified lake. Uh, so we have seen about stratified lake in almost 2-3 weeks so far. So you know that in a stratified lake there are going to be different zones in which different bacteria with diverse metabolic capabilities are going to be present based on the electron terminal electron acceptor and electron donors available in each of the particular stage. So stratified lakes pairs with environmental gradient niche. And finally, talking about consortium is cooperative microbial interaction. Consortium is a group of microorganisms, not a single microorganism. It's a group of different species and different classes of microorganisms that are in kind of a mutually benefiting relationship. So that's why we call it as a cooperative microbial interaction, wherein uh, the presence of one microorganism benefits the growth of the other microorganism and vice versa. So these are the answer for this match the following question. I hope it was clear. If there are any doubts, please let me know. Okay. Again, a repetitive but simple question. The algal partner receives dash and fungal partner receives dash from the symbiotic relationship in a lichen. I'll just wait for a couple of minutes and then we'll see the answer. Okay, so the answer for this is the alga partner receives water and nutrients, and the fungal partner receives its food from the symbiotic relationship. Okay, so the next question is the variable depth and area of nutrient availability in a stratified lake enables the presence of what? This is kind of an open-ended question, so whatever relevant terminology are able to correlate, you can just go. Any guesses? So, because there are different kinds of microorganisms, each not disturbing one another in any of the um, stages of the stratification. Let's call them stages. There are different levels, right? So we call them stages for our context. And in each of the stage, there is a particular kind of microorganism that can utilize certain resources, electron acceptor, electron donor that is present in that particular condition. The energy source as well, carbon source, energy source that is usable at that stage is being used by the right variety of microorganisms present in that stage. But when you talk about the entire stratified lake, there are different microorganisms that like occupy different stages and they coexist. Hence, this stratification allows a consortium to be formed and be present in that environmental condition. Okay, so we talked about foregut fermentation, hindgut fermentation, and there is also midgut fermentation. Which of these does ruminant animals undergo?
Microbial fermentation. Microbial fermentation.
Fermentation indeed is carried out by microorganisms. Yeah, but the question is about whether it is foregut, midgut, or hindgut fermentation. So that's why it is that blanket. goats, all of these are called as ruminant animals, and they have a four-chambered stomach. So the four chambers include the rumen, reticulum, omasum, and abomasum. Okay, so what happens is, since they are ruminating, which means they are chewing the cud, which in general terms we define rumination as. They are going to eat the partially fermented food, and this partially fermented food is being fermented in the rumen itself, which is called as the foregut. So the ruminant animals undergo foregut fermentation, and after that, the food travels to the other chambers, and the digestion process completes. So some examples of hindgut fermenters are horses and rabbits as well. So these are hindgut fermenters, whereas ruminant animals are generally foregut fermenters. Okay, now some questions on virology. So viruses are counted by PFU and bacteria are counted by CFU. You just have to tell if the statement is true or false.
What is the question here?
The question is whether the given statement is true or false. So the statement is that the viruses are counted by PFU, a unit called PFU, and bacterial counting we do in terms of CFU. You just have to figure out what is PFU and CFU and then tell if the statement is true or false.
Bacterial count are correct. That is colony forming unit. But
Exactly.
I don't know about PFU.
Okay. We just wait if someone else wants to answer, and then within a couple of minutes I'll just tell that.
Okay, ma'am.
Okay, so as rightly told by one of you, PFU stands for colony forming units, and mostly if we have learnt about or performed some microbiological experiments, you would have known what a CFU is. So basically, we take a sample and we prepare something called a media, which is the growth medium or the nutrient medium which supports the growth of the microorganism, and we try to dilute the sample and spread it onto a solid medium which contains a solidifying agent called agar along with the other nutrients. So when we are going to do this in a petri plate, which we call it as plating, then we find different colonies of bacteria growing, and we count the colonies and interpret the results in the in terms of CFU, which is the colony forming units. But when we talk about viruses, there is a terminology associated with virus estimation as plague forming units, which is abbreviated as PFU. So PFU is the number of infectious virus particles that are present in a sample. So what typically happens is we try to grow virus in a layer of host cells. Okay. So because virus needs another cell for replication, it cannot replicate on its own. So viruses are grown in a layer of host cells, and when this virus is going to infect that cell, obviously it is going to uh, hijack the entire system of the host, and it will try to replicate, and finally there is going to be cell lysis. Okay, so when there is going to be cell lysis and we have a layer of cell, we can spot at which place the cell has lysed, and there is a gap in the entire layer. So this spot we call it as a plague, and henceforth we can count viruses you in terms of plague forming units. So hence this statement is completely true. Viruses are counted by PFU and bacteria are counted by CFU. Okay, I just messed up the alignment of the slide. Anyways, I'll just go about. So here the assertion is viroids can encode proteins because they consist of naked RNA molecules, and here both the assertion and reason is true. So any idea what are viroids? We have not started with that yet. So yes, viroids are plant pathogens predominantly. Okay, here the assertion is that viroids can encode proteins. The reason is they consist of naked RNA molecules. So viroids are generally containing circular single-stranded RNA, and they also don't have a protein coat. Okay, but they cannot encode protein. So here the assertion is false and the reason statement is true. So the viroids do not contain a protein coat, henceforth they are naked, and they consist of a single-stranded RNA as their genetic element. So they are naked RNA molecule consisting entities, whereas the viroids cannot encode protein.
Yes.
Again, a true or false question: is biomagnification a prerequisite for bioremediation? this statement is true or false. Okay, let me explain what is biomagnification and what is bioremediation. So biomagnification, as the name suggests, something is going to increase, like magnify. Magnification in the sense there is going to be a exponential increase or a large amount of increase in something. So in biomagnification, we talk about the concentration of toxicants or pollutants. So toxicants or pollutants are generally some chemical compounds. Uh, let's take some pesticides for example, which all of us could relate. So pesticides are used in agriculture, but they are again harmful. They are again toxic substances, and there is a limitation of how much of pesticides and in what form should they be present in the environment for them to not cause harm to any micro or macroorganism, or even the it should not affect the chemical equilibrium of the place that it is present in. But what happens in biomagnification is, when this toxicant or a pollutant is generally present in a system, organisms tends to consume that, and we are aware of the food chain, food web and stuff. So it is just a sequential process where um, herbivores consume plants, and then comes uh, the plant eating, sorry, and then comes the animals that can eat the plant eaters, carnivores, omnivores, all of it, right? So when we talk about this food chain or food web, there is going to be constant consumption, and this pollutants or toxicants can get accumulated in one organism. Okay, let's say there is a small fish and a lake or a sea. Let's talk about environment. Okay, there is a small fish, and the water body is heavily contaminated with some metals or metals, let's say. So when the phytoplanktons are being produced, the phytoplanktons and the toxicant, everything in the water body, and the small fish will feed on all of it, and there is going to be accumulation of this metal in the small fish body. When there is even a larger fish that's going to consume this, then again the toxicant is being transferred from the small fish to the large fish, and when humans try to eat the fish, then we have birds that can eat the fish and the humans that can eat the fish, and this toxicity or the toxicant is getting transferred from one organism to the another up the food chain, and also the concentration also tends to increase. Why the concentration increases is, a bigger fish will not eat only one fish, one small fish, right? So it tries to consume multiple small fishes and accumulates all the toxicants that are present in all these fishes. So the concentration also tries to increase along the hierarchy of the food chain. So this is one of the serious problems in the environment. When we talk about bioremediation, this is the other extreme part of the conversation, which talks about the treatment process, where we can naturally use some biological method, like organisms, microorganisms, or macroorganisms like plants, all of this, or enzymes or biocatalysts for getting rid of the toxicants or the pollutants. So, if the question here is whether biomagnification is a prerequisite for bioremediation, I hope you are clear now. The answer is false. Biomagnification is not a prerequisite for bioremediation. Biodegradability is the prerequisite for bioremediation. So, if a pollutant is biodegradable, then we can go for some technique to biologically remediate that pollutant. In other cases, we cannot, like when we talk about metals can't be broken down by microorganisms, but yeah, they can be converted into other forms. But when we talk about some organic compounds, then microorganisms in itself can mineralize the toxicant or pollutant. So I hope the explanation about biomagnification as well as remediation was clear, and you're convinced with the answer for this question. If there are any doubts, please post it in the chat box. I'll repeat again if needed. In the meantime, the next question is, Again, a true or false question. One way to treat acid mine drainage is to add a reducing layer to reduce the sulfate and prevent reduction in the pH. Is this true or false? So, this is just an application oriented question. You just have to think a little and then answer. guesses for this, whether the statement is true or false? Okay, so the answer for this is true. So when we talk about acid mine drainage and when we correlate it to bioremediation, we try to add a reducing layer. So where we try to add some sulfate reducing bacteria there. So what happens is the sulfate reducing bacteria converts the sulfate to sulfide, and this helps the metals therein to precipitate. Okay, and in this process there is generation of bicarbonates and also consumption of protons, which again neutralizes the acidity in this process and raises the pH. So this is a typical acid mine drainage bioremediation process. Uh, so when we are adding a reducing layer, which primarily consists of reducing bacteria, the which is a sulfate reducing bacteria, sorry, I mispronounced. So it prevents the reduction in pH by consumption of protons and the generation of bicarbonates. So this statement is true. The next statement again is a true or false statement. You just have to tell if commensal bacteria are found only in the biofilm. Rightly mentioned that it is false. So commensal bacteria are not only present in uh, biofilm, they can be free floating organisms as well. And commensal bacteria, like to very commonly name, are present on the skin, the GI tract, all of it in the human. So they are not restricted to biofilm. And when we talk about what is a commensal bacteria, they can live on or within a host organism. Like it's very important to know that they can live on the surface or within the host structure itself, where they do not harm the host organism. So this is what we call as commensal bacteria, and they are not only restricted to biofilms. They can freely exist in other environments as well. Easy question probably, just to get all of you know about this. Again, a true or false statement: viruses are neither living nor non-living. Just give a true or false and then we'll move to the next question. I thought the question was easy. So yeah, viruses are neither living nor non-living because they possess genetic elements, but they do not have any membrane organelles as such, and they require a host organism for their replication and other processes. A living host system they require for like a bacteria or human or whatever plants, whatever they require. So they are neither living nor non-living. Okay, next one is a match the following question about different aspects of virology. So these are different components of a virus. You just have to match them. used for viral DNA insertion. So integrase is going to integrate something. A part of this viral DNA is going to be inserted into the host DNA, and that is done by the enzyme called integrase. So hence it pairs with viral DNA insertion. I hope it was clear. Again, a open-ended question. You can answer whatever you want. So, what virus is defined as what? So, it can differ in terms of each of your perspectives and what you know about virus. So, what is a virus defined as? Viruses are neither living nor non-living. They are infectious particles, and they can also be called as subcellular genetic elements. This is just to get you introduced to this word. Okay. Okay. What is a virus without a protein coat called as? Okay, a virus without a protein coat is called a viriod or a extracellular virus particle. Uh, Name the hypothesis related to viral evolution. There are three hypotheses. You can name any of them. so the three hypotheses related to viral evolution are alternative hypothesis, reductive hypothesis, and Michael-based evolution hypothesis. So the alternative hypothesis states that the genetic fragments that code for essential cellular functions escape and gain viral genome. These genes replicate in cellular host and form virus-like particles. Okay. So what I am trying to tell is there are essential function determining gene fragments. They get integrated to the viral genome and then they give the access for the virus to go and survive with the help of a host. And in reductive evolution hypothesis, they talk about primitive cells with reduced size and genome get encapsulated by endosymbiont and develop into viral-like particles. There are primitive cells, they get encapsulated by endosymbiont. I think you might have heard about this endosymbiosis theory where uh, some organisms are encapsulated by another organisms and the both together start functioning and the other becomes like a organelle, something of that sort. Is the reductive evolution hypothesis. In the micelle-based evolution, so the primitive atmosphere had micelles, and these micelles could have trapped the nucleic acid, and over time they also had the ribosome activity, and they tried to form the structures called protovirus. So these are the hypotheses that are related to viral evolution. Okay, again a true or false question, whether the mutation rate is directly proportional to the genome size. This statement is again false. Mutation rate is not proportional, directly proportional to the genome size. In some observations, they have observed the opposite, where it is inversely proportional. And henceforth, they try to suggest that the mutation rate remains the same only. Based on the genome size, the effect gets magnified or non-magnified. So that is the point here. There are a few more questions. I'll just walk through the questions because we are short of time. I'll not wait for you to answer. I'll just post the question and answer simultaneously. So a virus with a capsid and a nucleic acid only are called naked viruses. So they have a capsid and nucleic acid. There is no coating for the nucleic acid, and they are called the naked viruses. Viruses that can switch from single-stranded RNA to double-stranded DNA are called retroviruses. This you might have studied. So they use this reverse transcriptase for converting. from RNA to DNA. So hence they are called as retroviruses. And the viruses that can switch from double stranded DNA to single stranded RNA are called hepadna viruses. So retroviruses, maybe you might have heard, because HIV is a retrovirus, and there are a few other common examples. And when you talk about this reverse transcription also, you would have come across, but there is one more interesting phenomenon in viruses where uh, hepadna viruses can switch from being double stranded DNA viruses to single stranded RNA viruses. Okay. And the next question is viral number increases in latent period of cell cycle. So we just have to see if the statement is true or false. So in the latent period in viral replication, it this is the phase that immediately follows the infection. So once the virus has entered the host cell and it has not begun to produce any new viral particles, that in between period is called the latent period. So in this time there is no increase in viral number. Only the viral genome gets replicated, and the assembly of the new virus particles have not yet uh, started. So hence this is a false statement. So I hope you understand what is a latent period. It is a phase where the virus has infected the host cell, but new viral particles have not yet started to produce. So this is the latent period, and in this process only the genome could have multiplied, but there is no new viral particles that are on the host generated. So the viral number does not increase here. And similarly, we have seen about transformation, transduction, and all in one of the slides in previous session as well. So in transduction, the genes are transferred from the donor to the recipient by with the help of bacteriophagy. So in last class we saw about horizontal and vertical gene transfer. We saw what is transformation. So we saw the use of pili and stuff. So in transduction, there are just phages that help in this transfer of the genetic material from one organism to another. So yes, this again, if you have studied about some viral microbiology, you could have known the answer. Like, this is very repetitive. Viral replication can be lytic or lysogenic. Lytic means lysis is going to happen there, just like we saw in this plague forming unit characterization as well. Once the microorganism, sorry, once the virus is replicated inside the host, which can be a microorganism, the cell can rupture. So this is the lysis process, and in lysogenic process there is no cell rupture, but it gets uh, transferred to other organisms without the help of cell rupture. So yeah, this is again what we saw extensively. Enzymes present in retroviruses are called reverse transcriptases, which can convert RNA to DNA. And polyoma viruses cause tumor. So the viruses that cause tumor are called polyoma viruses. Again, this question is regarding mutation DNA and stuff. So here uracil is a RNA specific base, and it's thymine in DNA. Okay, and for DNA we have this component makes the DNA stable. So it's going to be uracil because of the presence of the difference between DNA and RNA is one is it is single stranded, and the other is RNA has uracil whereas DNA has thymine, and this also determines the stability of the molecule, and thymine confers the stability to DNA in comparison to uracil not conferring such stability to RNA. And reverse transcriptase again converts RNA to DNA, and capsid homology stands as an evidence for viral evolution. The capsid has been homologous in multiple viruses. So henceforth, when people study about this viral evolution, they need a point where convergence is there, where divergence is there, to predict how this evolution, or to track down the direction of the evolution. So capsid homology is one aspect that gives us information about the viral evolution. And finally, we have the genome size. So genome size tells us about the mutation rate determination. So we saw in one of the questions whether it is directly proportional or inversely proportional. So it determines. And anti-protozoan drugs can attack humans also. Whether the statement is true or false? Yeah, the statement is true, because protozoans are eukaryotes, and henceforth a drug that is designed to attack eukaryotic cells can also attack humans. Enteric protozoa require vectors. So enteric protozoans can directly come in contact through ingestion. They don't need vectors to get transferred. Hence, the statement is false. So finally, what is lichen we have seen, and this slide also we have seen already. With that, I would like to close today's session. If you have any doubts, post in the chat box, or please come back to the next session and ask. Thank you for joining in for today. I hope the session was useful.